Okay, so this is chapter 3, part 2. And so again, we're talking about biological macromolecules. And our focus for this part of the chapter is going to be on carbohydrates. And so again, carbohydrates are large macromolecules. They are polymers. So they are large molecules made up of repeating subunits, and those subunits, monomers, in this case will be simple sugars, which we also call monosaccharides, or saccharides, meaning sugar. Carbohydrates are extremely important to our diets. The recommended daily allowance for adults for carbohydrates, they should make up about 45 to 65% of our total calories. And that number varies depending on age and gender and weight. Good sources of carbohydrates include grains, fruits, vegetables, legumes like lentils, dairy products, and then you could also get carbohydrates from sugars and sweeteners like honey, and processed foods like pasta and white bread. But again, there are better choices. So whole grain foods, fruits and vegetables, fresh fruits and vegetables, legumes, good choices. And then of course there are others, but some are better than others. Carbohydrates are important. They're broken down into glucose monomers, and glucose is the fuel source for your brain. Your brain depends on glucose to be able to function. Okay, so very, very important. Okay. And so again, saccharides meaning sugar. And so carbohydrates are going to contain carbon, hydrogen, and oxygen atoms. And they're going to be in a specific ratio of 1 to 2 to 1. So for every carbon, there will be twice as many hydrogens and an equal number of oxygen. And then this little n outside of the formula here means, this can be anything. If this was 2, then there would be 2 carbons, 4 hydrogens, and 2 oxygens. And so whatever this number is out here, you have to multiply that by the number next to each of these elements. And so next to carbon is a 1. We don't write it, but it's assumed that it's there. And next to oxygen is also a 1. So this is always going to be the ratio for all carbohydrates, no matter how large or small they are. So carbohydrates are going to be, again, simple sugars, and then complex carbohydrates will be polymers of sugars. And so we have really 3 main types. So we have monosaccharides, which are composed of just one sugar, and we'll look at those. And here's an example here of glucose. And then we have disaccharides made up of 2 sugar units. Okay, and so here's an example here, right there. We'll talk about those. And then we have polysaccharides. So polysaccharides are going to be made up of many sugar units. And so here's an example down here of a polysaccharide. And then I just wanted to mention oligosaccharides. And so here's an example of an oligosaccharide. This is a sugar called raffinose. It's made up of 3 sugar units. And again, I'll mention those during the presentation as well. Okay. So let's start off with the simplest of the sugars, which are the monosaccharides. And again, these are made up of just one single sugar, one monomer. These are going to be the monomeric building blocks, the subunits, that'll be put together to form disaccharides and oligosaccharides and polysaccharides. Okay? These simple sugars can range between 3 and 7 carbon atoms long. And when we look at each carbon atom, each carbon is going to be bonded to a hydroxyl group, which remember is an OH, an oxygen, and a hydrogen, except one. That one last carbon is going to be double bonded to an oxygen atom. So we're going to have a carbonyl group in the molecule. And so pictured here is the cyclical structure. And so we're going to learn that monosaccharides that are longer will tend to form a ring structure, especially in aqueous, watery solution. So glucose, which is C6H12O6, look at that ratio of carbon to hydrogen to oxygen, right? For every carbon, there's twice as many hydrogen, and an equal number of oxygen. That's always going to be the same. So glucose is the most abundant monosaccharide on the planet. And again, it's used as the primary energy source. And so when we talk about cellular respiration, we're going to do that using glucose. Okay, and remember again that glucose is going to be the primary energy source for your cells, especially your brain cells. It's not the only source of energy that your cells can use, but it's the preferred energy. So here's some examples of monosaccharides. And so here's an example of glyceraldehyde. Glyceraldehyde is classified as a triose, meaning 3 carbons long. And so look, 3 carbons. 2 of these carbons are attached to hydroxyl groups, here and here. And that last carbon is double bonded to an oxygen. So here's my carbonyl. Those functional groups are coming in handy from the last chapter. So here's a second example. This is ribose, and we're going to learn that ribose is actually the sugar found in RNA, which is a nucleic acid. We'll get to that later on in the chapter. Ribose is a pentose sugar, so pent meaning 5. 5 carbons, 1, 2, 3, 4, 5 carbons long. 4 of those are attached to hydroxyl groups, this OH, and 1 of them is a carbonyl, double bonded to an oxygen. And here's glucose in the straight chain form. Okay, so again, these longer sugar molecules, so ribose and glucose, these are both going to form rings. They're going to cyclicalize in aqueous solution. But here it is as a straight chain molecule. And so glucose is a hexose, so 6 carbons long. We got, start from the bottom. Doesn't matter which way you start. Actually, it does. So 1, 2, 3, 4, 5, 6 carbons, right? And so 5 of those carbons are bonded to hydroxyl groups. And this last carbon up here is going to be double bonded to an oxygen. So there's that carbonyl. So those are 3 examples of monosaccharides. Okay, here's a couple more. We already saw glucose, but look, here's galactose, and then here's fructose. And I wanted to show you these because I wanted to show you that these are isomers. Remember that term from last chapter? Isomers are going to be molecules with the same molecular formula. So in this case, C6H12O6, but they have a different structural arrangement. And so you can see fructose has its carbonyl group here on this carbon instead of this carbon here. And if you look at galactose and compare it to glucose, it's carbon number 3. Carbon number 4. In this case, it's where the hydroxyl group lies. So it's to the left of this carbon here, and it's to the right of this carbon here. Okay, so those are isomers. Okay, so again, when you put sugars into aqueous watery solution, if they are longer molecules, so 5, 6, 7 carbon long, they're going to tend to form, they're going to cyclicalize. They're going to form a ring structure. And that's because that's a more stable conformation for the sugar. And so you can see this is glucose, right? So glucose, we just saw it as a straight chain. And when it cyclicalizes, we have this carbon here, which is going to bond to this oxygen. And so we end up with glucose, ring structure. There's actually 2 different forms of glucose, and it all depends on how this reaction happens. In this case, this is alpha glucose, and this is beta glucose down here. What's the difference? So if we look at carbon number 1, okay, if this reaction occurred in a way that the hydroxyl group on carbon 1 falls below the ring, it's alpha glucose. If it's above the ring, it's beta glucose. Okay, so there are 2 different forms, and that is important. These 2 different forms will form different polysaccharides, and so we'll see that a little bit later. And then I wanted to show you 2 different, the 2 other sugars. So ribose here and fructose. Again, these are 2 monosaccharides. And again, we saw the linear structure. And now here we are, we see the ring structures. And so remember, again, ribose we're going to come back to when we talk about DNA. Okay, so those are monosaccharides. So let's talk about disaccharides. So di meaning 2. So a disaccharide is 2 monosaccharides that get put together. The process for putting them together is called dehydration synthesis. which again we saw in the previous part of this chapter. So it's the same thing. Water gets removed and a covalent bond between the two monosaccharides is formed. Okay, so here is glucose and here is fructose. Okay, these are actually isomers of each other. So we've got two sugar molecules, two monomers. We're going to take out water, and this time the right things are colored. So we have this red OH and this red H. So we're taking off a hydroxyl group and a hydrogen. We're going to pull that out as water, so not shown here, water gets removed. And then this oxygen will covalently bond to this carbon, carbon number 1. Notice the numbers on the carbon. So in glucose, this is carbon 1, 2, 3, 4, 5, 6. Okay? In fructose, it's a little different. 1, 2, 3, 4, 5, 6. So carbon 1 is going to be linked to carbon 2 of fructose. And so that linkage, this bond that is formed, is called a glycosidic linkage or a glycosidic bond. That's what we call all the covalent bonds between sugar molecules. So if we have a disaccharide or a polysaccharide, all of the covalent bonds in between two monomers, they're always called glycosidic linkages or glycosidic bonds. Now this is an anabolic, a reaction that we're building a bigger molecule, and so this is going to require ATP. It will, of course, require enzymes because all metabolic reactions are catalyzed by enzymes. Okay? And so I just wanted to give you a couple of examples of monosaccharides. And so the first one pictured here is maltose. We saw this in the previous part of the chapter. And so maltose is a disaccharide made up of two monomers of glucose. So here's glucose and here's glucose, and they're linked together here by a covalent interaction, which we call the glycosidic bond. Lactose is this second one down here. Lactose is made up of glucose and galactose. Lactose is a sugar found in milk, and some individuals lack the enzyme to actually break this down. And so if you are lactose intolerant, meaning that you have an intolerance for this sugar, you lack the enzyme lactase, which is responsible for breaking this sugar down. And so because you can't break it down, it ends up passing through your gut and ending up being broken down by the bacteria in your gut instead of you. And so these bacteria will produce gas as a byproduct, and you end up with cramping and gas from that. Okay, not all individuals are lactose intolerant, but some people are. The last example down here is sucrose, which is common table sugar, the white stuff that you might put in your coffee, and that is also a disaccharide made up of glucose and fructose. And again, I just wanted to quickly mention oligosaccharides. These are polysaccharides. They're made up of more than two sugars, so they're technically a polysaccharide. But oligosaccharides, they're usually just a small number of monosaccharides bonded together, usually 3-ish. And they're an intermediate between disaccharides and polysaccharides. I wanted to mention these because they are important cellular components. They're going to be attached to proteins and lipids to make what we call glycoproteins and glycolipids. And so anytime you have carbohydrates attached to a protein or attached to a lipid, that's the glyco. That means carbohydrate. And so these are going to be important in things like cellular recognition and cellular attachment. And so when we talk about in the next chapter, cell structure, we'll mention glycoproteins and glycolipids. So I just wanted to show you that here. The example that's here is raffinose, which is an oligosaccharide made up of 3 sugar units, galactose, glucose, and fructose. And raffinose is found in foods like beans and broccoli and cabbage. And this is another one where some individuals will end up with cramping and gas because, again, they lack the enzyme to break this down. And instead of being broken down by our enzymes, just like lactose, it's broken down by gut bacteria, and gases are produced as a byproduct. Okay, so let's move on to the big guys. So these are polysaccharides. These are our complex carbohydrates. Okay, these are long chains of monosaccharides. Again, linked together through dehydration synthesis reactions, and in between these monomers are going to be glycosidic linkages or glycosidic bonds. Polysaccharides might be branched or unbranched. They may contain the same monosaccharide, or they may contain different monosaccharides. And we're going to talk about a lot of different ones. Mainly these polysaccharides are either going to be classified as energy storage molecules or involved in structural support. And so we're going to talk about 4 examples. We'll talk about starch, glycogen, cellulose, and chitin. Those are our 4 examples of polysaccharides. And so here is our first one, and this is starch. So starch is going to be found in plants. This is the storage polysaccharide in plants. So this is how plants will store energy. Okay, they're going to take glucose monomers and link them together through dehydration synthesis to form starch, a polysaccharide. And then they'll store this starch in special locations. So for example, in this example, this is a microscope image of plant root cells. And so all this purple stuff in here is starch. So we've actually stained this so that that shows up. And so starch gets stored in roots, tubers, and seeds. When the plant needs energy, it will go to these reserves and it will do hydrolysis, and it will break those sugars apart, individual sugars, and use those sugars in cellular respiration. There's 2 types of starch that plants have, and they just differ in their structure. So amylose and then amylopectin are the 2 types of starch. Both are made of glucose monomers. Amylose tends to be a straight chain molecule, and amylopectin is branched, so you can see it branching out. That's the only difference between the 2. Okay, so plants store glucose in the form of starch. Animals, like humans, we also store glucose. Okay, but instead of storing it as starch, we store it as glycogen. Okay, so that's our storage molecule for polysaccharides. So this represents a glucose store in animals. It's an energy reserve that we can go to. Okay. Glycogen tends to be highly branched. So I was looking for my image. So here it is right here. So you can see all the little sugar, all the little glucose monomers. It is made of glucose. Okay, and they're going to be branched, not straight chain, highly branched. Glycogen gets stored in the liver, which is shown here, as well as muscle cells, which are actually in this picture here. And you can actually see these little granules here. These are glycogen granules, and these are lots of mitochondria. So your muscle cells are chock full of mitochondria because they're doing cellular respiration, right, breaking stuff down to make energy for movement. So when your glucose levels in your blood run low, what's going to happen is your liver or your muscle cells are going to be triggered to undergo what's called glycogenolysis. And that's going to be, again, when glucose levels are low in the blood. And what happens during glycogenolysis is that glycogen's broken down and glucose is released. So this is a hydrolysis reaction. So water is required, bonds are broken, energy is released. Okay? The breakdown and synthesis of this glycogen are going to be tightly regulated, right? So when blood glucose levels drop, you do glycogenolysis. When blood glucose levels go up, glucose stores are going to be made. Okay, this is tightly regulated, right, to maintain homeostasis. The hormones insulin and glucagon are going to play key roles in controlling glycogen metabolism. Okay, and we're back to plants. And so starch is an energy storage molecule. Cellulose, which is also only found in plants, is going to be a structural polysaccharide. Cellulose is the most abundant polymer on the planet. It is the main structural polysaccharide found in plant cell walls. Okay, so, oh, this image got a little fuzzy. The plant cell wall is going to be the outermost structure on the plant cell. And if I were to take a look at this with an electron microscope, I would see all these overlapping fibers. And those are cellulose fibers. If I was going to look at each one of these individually, this is what I would see. So cellulose fibers are these long chains of glucose. So here are the glucose monomers all linked together by glycosidic linkages. And notice that each glucose monomer is flipped compared to its neighbor. So if you look at this glucose, right, the CHO2 group, we'll say, is on the bottom here. It's on the top here, and then it's on the bottom, and then it's on the top. And it would keep going like that. And the reason for that, this flip-flopping, makes this cellulose fiber really straight and really rigid. And that's what we want, because this is a structural polysaccharide. Okay? That helps to maintain the rigidity of the cell wall. These can stack right on top of each other, all these fibers, they can crisscross and stack, and you end up with a really strong cell wall. In animals, right, when we eat plant material, we can't break down cellulose. It's indigestible. And so this is our fiber in our diet. Okay? And fiber slows digestion. Fiber helps us to feel full, and fiber helps us to go to the bathroom. So there's lots of benefits to fiber. And so eating a diet rich in plants, so fruits and vegetables, is really, really important. Now there are some animals that have formed symbiotic relationships with other organisms to help them to actually digest cellulose. So for example, cows, right? Cows feed on grass and hay. Cows can digest cellulose. They can't actually digest it themselves. They don't have the enzymes, but they have gut microbes that can. Same thing is true for like termites, right? Termites feed on wood, which is just cellulose pretty much. Termites don't have the enzymes to digest cellulose, but they have gut microbes that can. So they've got these symbiotic relationships that allow them to eat plant material, cellulose, and actually extract nutrients from it. Humans do not have that capability, so for us, cellulose is just fiber. Still important, but not digested. Okay, and lastly, chitin. So there are some animals that have the equivalent of cellulose. But we don't call it cellulose, we call it chitin. So chitin is another type of structural polysaccharide. It's present for structure and protection, right? So it provides structural support, and it also provides protection to these organisms. It's a different monosaccharide here. They're long chains of what's called N-acetylglucosamine, which is different than what we've seen so far. It's another monosaccharide. Where do we find this? So any arthropods, like the buzzing bumblebee here, insects, crustaceans, they're going to have an exoskeleton, which is going to be made up of chitin. Okay? And organisms that have this type of exoskeleton will go through a process called molting, where they actually have to shed their exoskeleton as they grow. Okay? We also find chitin in the cell wall of fungal cells. So not just animals, fungi as well, which is pretty neat. We actually use chitin. We will use this material medically and in industrial applications. So if you've ever had surgical sutures, they're made of chitin. There are wound dressings that are antimicrobial that are made of chitin. When we engineer tissues in the laboratory, the scaffolding used for that engineering is usually made of chitin. So there's lots of applications for that. So that concludes the part of the chapter on carbohydrates. The next part of our chapter will be on lipids.